Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
Welcome everybody to the first SBGrid webinar of the academic season. Couple process things first. So this is our first webinar. Our next webinar is going to be November 18th, and that's going to be Hui Wang from the Zoo Lab in UCLA to talk about TomoNet. If you have ideas for future webinars or things you'd like to see, then feel free to let us know at events@SBGrid. Throughout this webinar, if you have questions, feel free to either use the Q&A feature or send them to one of the hosts in the chat. And with that, I'd like to introduce our presenters today. Alisia Fadini, who is currently in Columbia and formerly in University of Cambridge, and Minhwan Lee, who is currently at the Flatiron Institute and formerly in Hextra Lag at Harvard. So, and they're here to talk about Rocket, which I found very cool when I was reading through the paper, and so I'm looking forward to this. So with that, I will not belabor the introduction and turn it over to you two. So thank you for joining us.
No, thanks. Yeah, thanks a lot for having us. We're actually very excited to have the chance to participate in this webinar and make Rocket available through SBGrid. It's quite new, so we'll maybe introduce it and hopefully walk everyone through what it's like to start using it, and hopefully we'll get some feedback from the community, given that it is quite new on our end. I think what we've decided to do is that I will just give a brief overview of the motivation and the kind of core concept behind Rocket, and just like illustrate a couple of examples of where we've seen that it can be useful. And then Minhwan will kind of take it over from there and actually try to show in practice what it's like to try to use it with your own data. So I think without further ado, I will start on my end. So hopefully you can just see my presentation.
Yep, slides look good.
Great. Okay. Yeah. So I'm Alisia. Minhwan is here with me. We have been working on essentially trying to guide protein structure prediction methods, particularly kind of our open source version re-implementation of AlphaFold 2, which is OpenFold, through experiments. And we've called this program Rocket, which you'll actually see in a little bit actually stands for something. So I guess like the thing I don't want to be kind of lost at the end here is that this has been actually a fantastic collaboration. We're very thankful to have been a part of it. I did most of this work when I was in Cambridge in the past year and a half with Randy Reed and Early McCoy and the Phaser team, and also kind of jointly with Mohammed AlQuraishi's group at Columbia. Duke was with Minhwan was with Duke Hextra at Harvard. We've had a lot of support from the Phoenix Consortium, the OpenFold Consortium, the reciprocal spaceship developments. And of course, now we're very happy to have Rocket kind of becoming available through SBGrid. So these are, I think, like really important acknowledgements that made the work possible. And the motivation for kind of developing Rocket really was a little bit in this like context of I think an exciting time for structural biology, where we have a lot of the data sets that we've had access to for a few decades now, but also like new and exciting data sets, both in throughput and in what we're able to see. So we have more and more kind of observations of transient complexes, of reaction intermediates, and also of, for example, through cryo-electron tomography, observations of like our proteins and structural observations within the context of the cell. And then what we've been trying to look at is the step of going from the data observables that we collect to an atomic model that we might be able to interpret mechanistically. And I think actually there's been a lot of progress both on the cryo-EM side and on the crystallography side, but we do still have like a few tasks that remain challenging. And I think to name two that are big, at least in our view, are on one hand like high throughput automation in the context, for example, of large fragment screens and obtaining structures, refined structures in this context. And then of course we still struggle when the data is lower resolution, either for kind of hardware reasons or because we have disordered regions or multiple states overlapping. And I think actually like quite a few others in the field, we started wondering with these new machine learning models that have become available that can accurately predict protein structures, can machine learning help? And I think also, working with Randy and molecular replacement, there's one aspect in which we can definitely say that it already has significantly helped, and that's molecular replacement. So we can now almost always solve the phase problem through using an AlphaFold prediction. Here I'm showing AlphaFold 2, but AlphaFold 3 is similar, and kind of like having this as an initial model to use for our data, to either dock or phase to our data. But still, we always need that kind of extra step, right, where the actual predictions that come from these models are not accurate enough in many of the details that we actually care about from like a chemical and a biochemical perspective. And so we have this extra step of then fitting the initial prediction to the density or the map. And you could imagine, it's something that we don't really have yet, but you could imagine something that I've kind of vaguely called here the ML refiner, where you could have a structured prediction algorithm that starts with a sequence information, but also with a set of condition-specific observations that you've obtained from an experiment to predict either a single like protein or macromolecule conformation, or ideally even like multiple conformations that can like fit and explain this data. And I think more on just like this kind of streamlining and automation, there's also an interesting perspective here, which is that these models, they have inherently kind of learned and encoded different ways of representing protein structure, which could, and this is a hypothesis that we started testing, that could perhaps help us in some of the difficult model building tasks where traditional geometric restraints sometimes fail. So what Rocket is kind of a first attempt at streamlining the single, because we're focused on AlphaFold 2. We started this about a year and a half ago when AlphaFold 2 was really the main model that we wanted to try to rely on. And so it's a first step at trying to go to kind of automate this process of using both experiment information and sequence information to get to at least one main conformation that explains the data. So the concept is that we condition predictions from AlphaFold 2 with experiments so that an initial prediction that might not fit the experimentally collected data then actually is turned into a prediction that maximizes fit to the data. And right now, Rocket works with either X-ray intensity data or structure factors and cryo-EM half maps. So the core idea that we wanted to try to leverage when we started brainstorming on how we wanted to go about this was, on one hand, that we wanted to start leveraging an existing architecture without requiring extensive retraining or training. And on the other hand, of course, there's like a computational and resource reason to do this, but there's also the fact that if we're not training and kind of committing to a specific data type, well, the approach that we end up developing and the framework that we end up developing is actually something that can then flexibly adapt to different data modalities. And so we don't commit to, for example, either only working with cryo-EM or only working with crystallography, but we can adapt to other data modalities as they become available. And so what we ended up converging on is this iterative inference time optimization, which sounds like a bit of like a mouthful, but is actually simple in concept. So what we do and what Rocket does is that kind of traditionally as with normal AlphaFold prediction, we start with an input sequence. And this input sequence is also used to collect coevolutionary information, so multiple sequence alignment. And then what we do is we run actually just pre-trained AlphaFold to really our open source reimplementation, so OpenFold inference to just obtain a structure prediction. And now, because we have this prediction and we have experimental data, we can just formulate a kind of a typical refinement target of what the likelihood of the data is given this initial model. And this becomes possible to kind of directly interface with the OpenFold framework if we have developed kind of a differentiable pipeline so that lets us maintain and do gradient backpropagation all the way through this process. And this is actually was a large part of making this happen that Min Huan worked on during his graduate work at Harvard. And so if we have this set up, then once we have a structure prediction, we can say, okay, how well does this prediction match the data? And then because we have like connected everything together, we can take gradients and we can go back inside an internal representation that AlphaFold has for the protein and say, okay, now update your representation so that not only you keep predicting a structure that you think is feasible, but also you are improving the likelihood to our data. And we can do this iteratively so that we iteratively predict more like kind of consecutively improved structures. And ideally, we would then converge. So a question here for us was where in kind of the monster that is AlphaFold 2 should we go and try to actually inject experimental information? We've outlined this in our preprint, and I won't go over it here, but the kind of key conclusion we came to is that we actually adapt and modify the coevolutionary information, this kind of continuous embedding that AlphaFold has for coevolutionary information. And so what Rocket does is it uses this initial embedding at first, just like the vanilla AlphaFold embedding. It runs through OpenFold, and then we have a loss function, a traditional likelihood loss function that accounts for error both in the model that we're currently building and in the measurement that we've acquired. And then through this kind of maximum likelihood target, we continuously update. All we do is a linear perturbation of this initial matrix. And we do this until we iterate. And this is really what Rocket is. So the main trunk that we'll be talking about is implementing this approach. So, and just one minor thing, we haven't yet incorporated B-factor refinement kind of as we go along. So right now, and we have some interesting results on this, we let this kind of pseudo B-factor that has also previously been used in Phoenix and Rosetta to reflect the confidence that AlphaFold has on each residue. So we have this pseudo B-factor that reflects PLDDT. So if the model is very confident, we have low B-factor, and as the confidence improves, as the confidence decreases, this like pseudo B-factor explodes. So at the end, usually of running Rocket, we automatically concatenate a Phoenix refine step that fixes the B-factors. And this is just a technical note because I know a lot of people here will actually be interested in using the tool. So we've looked at a little bit at like in what cases this could become useful, like perhaps model building challenges that are more underserved by other existing techniques. And what we've seen is like especially flexible loops that usually maybe get cut out or need like manual input, and low resolution and large domain rearrangements are kind of where we've seen that Rocket can really shine. I am just going to show a couple of examples. very, very briefly, just to kind of put some ideas out there. So one thing we've noticed is that doing this kind of refinement in coevolutionary space, rather than doing it in atomic Cartesian coordinates, as we normally would with, for example, RefMac or Phoenix Refine, is that the barriers that are required for specific structure rearrangements end up, we think, being reduced because we're moving in a different space. And so this is one example of a rocket trajectory I'm going to play, where we have a human able kinase that is predicted by AlphaFold in its active state, and it's this like kind of pink bubblegum protein, and I'm kind of showing the drug-bound inactive state that's like trapped in this transparent outline. And so you see, Rocket is able to kind of adapt this ring to match the experimental conformation and undergo some large changes, which normally would be more difficult to overcome in Cartesian coordinates. Another example I'm going to play here, and I just wanted to show kind of how this looks actually as in like gradient descent with the experimental loss, is a time-resolved example from serial crystallography, where again, I just maybe want to highlight this helix here. The blue is the initial AlphaFold prediction, and the gray is what the group ended up depositing in the PDB. And so again, like as we kind of improve in likelihood, you'll see that Rocket can kind of adapt and then like register shift this helix into place. And so some of these like larger changes now become possible by moving in this different space. The other thing we've seen is that it can be quite useful, especially for fold recovery at low resolution. So again, this is an example of a transporter protein that is predicted by AlphaFold, again, in a perfectly good conformation. It's just the inward open state, and we have a cryo-EM map of the outward open state. And we've done some reprocessing of the maps to kind of progressively obtain lower resolution data. And we've seen that Rocket can kind of up to 10 angstroms still recover this initial fold starting from, this final fold starting from the initial AlphaFold prediction. So I think to kind of wrap up a little bit this teaser introduction, like what have we learned so far from Rocket, is that by doing this different structure optimization that is in AF2's coevolutionary embedding rather than Cartesian coordinates, we can enable automation of modeling tasks that are usually more difficult, and that we do see robustness at low resolution, and we're happy to answer more questions about this. The key thing here is that we've implemented it right now with crystallography and cryo-EM. In principle, if you can formulate a log likelihood target for your data, then this is adaptable to kind of any other data type. And Min-Hwan will slowly kind of introduce our Git books and our documentation and our preprint. So a couple limitations. So I've mentioned this is AlphaFold 2. So right now we can only do proteins. So you can obviously have DNA, like I showed with the photolysis example, or a ligand there, but Rocket won't know about it. So it will only try to refine the protein component. And then again, our first prototype is memory intensive. And so at the moment, you kind of on a GPU will require around 500 and 600 residues at a time. So either like one chain at a time or one domain at a time in cryo-EM, for example. And we are actually working soon on making a server available through Phoenix, so that if you don't have GPUs available, you'll still be able to run Rocket by connecting to the server. So what are we hoping to work on soon? Actually, our first kind of work is on the improved memory requirements, and we're collaborating with a team on that to just make the process less compute and time intensive. And we'll start now extending this to the AlphaFold 3 architecture, so that we'll be able to handle like ligands and non-proteins. Eventually, and probably in a little bit, we'll try to also tackle multiple conformation. And again, we can answer more questions about this later. But I think now I'll kind of transition more to the practical side of like what if you actually want to try to run this. And so in practice, the rocket pipeline is kind of divided into a first pre-processing step. And this is where we do, this is actually a step that is dependent at the moment on Phoenix. And so we run some internal and Phoenix commands to do some error parameter estimation for refinement likelihood targets. So for crystallography, we do the sigma A and the kind of some error estimation based on measurement uncertainties for the intensities. And similarly, we use the two half maps. So we can work with only one map, but ideally we use the two half maps for cryo-EM to, again, have an anisotropic error model. And if needed, we can do a molecular replacement or docking. So this is happening in the pre-processing step. And then the default is usually to go to that kind of this main rocket trunk, which is like a simple rocket refine command. And we'll go through the details of what these inputs are in a second. There's also a kind of an optional pipeline that is a bit more time intensive. So we haven't made it the default, and it's kind of, you should probably use it if you're anticipating a large fold switch, or if you're really unhappy with the initial rocket refinement. And what this does is actually includes some MSA subsampling and clustering to kind of provide you with better initial confirmations. Well, with a larger variety of initial confirmations from AlphaFold, and then it will score each of these predictions to the experimental likelihood and provide you with the best scoring confirmation that you can then further refine with rocket refine. And again, Min will kind of talk through this in more detail. So I think we'll probably move to just the demo side, and we're going to kind of demo how you would go around launching this with your own data, with using a Serpin example that we also show in our preprint. And this is a high resolution data set, though, where the initial AlphaFold prediction needs to undergo a large fold switch, where this external loop needs to reinsert into the fold for this inhibitor-bound latent state of the Serpin. And then we'll show kind of similarly an example that's also in our preprint of a GRO-EL subtomogram average, where we recover the two chains from the two rings. And so I think with this, I'll probably pass the word to Min Huan, and then he can share his screen.
Can you all see my screen now, I guess? Okay,
Yep,
okay.
looks
Thank
good.
you. Okay, thank you for the invitation. I will thank you for the intro part from Alisia. In the following like 20 minutes ish, I will try to walk you through the two demo cases, as Alisia said before, about like one for X-ray with high resolution Serpin case, and the other from the cryo-EM part using the subtomogram. So like all the materials like I will mention like in this demo has already been uploaded to the nodal of repo we created. So like you can get access to the link from our repo or from our Gitbook Rocket Docs. You can find it here. And today, mostly I'm trying to like walk you through the two cases, like the Serpin case here, as also like listing our like docs, and also like specific about how you can launch your own X-ray data or cryo-EM data. So I will get started to say like what is the important steps and what is the file, or like what is the input file and what is output file you should expect from the rocket. So for the X-ray case, the basic target is like you have some experimental magnitude and you want to refine an atomic model which best match your experimental observables. So what you need for this whole pipeline is, first you have to prepare your input files as organized in the following way. So as I will show it here, we go to the X-ray case. And if we try to show what we have here. I would ignore this pre-processing because that is one including the output of the rocket. Yeah, so this is exactly what we need for the rocket input. So this MTD file will include what you have from your X-ray experiments, including the magnitude and the uncertainty of that observations, which are typical MTD files. And if you don't really have the TNG and with R-freeze, like just put it there, like the preprocessing will take it, will take care of that for you. And the pre-duct PDB is actually the models you believe is a good MR solution to your data. It's also fine if you don't have that. The preprocessed part will try to call the phaser from the Phoenix part to run the MR solution for you, but it might not be as good as you prepared that for it. And also we need a fast file with the sequence of the proteins of the target proteins. And also we need some like multiple sequence alignment pre-computed for these systems. And now we have to ask users to like pre-generate them from some servers outside of the rocket and to like organize them in this way. But now we are also working on a new like server which can call the one coming from MMSeq to really provide the MSA files for you on the fly. So now once we have that, you can now run the rocket preprocess command, which requires you to provide a few like arguments. The first one is the file ID, which is basically the one how you name your system. It should be consistent with the prefix of the folders here. As you can see, like I call it 1AirJ5, so that this one should be 1AirJ5. And this file ID will be used to try to find those PDF files, those MTD files, and all these like MSA files. And second, you have to tell it like if it's X-ray or cryo-EM. So for this case, we call it X-ray. So you just like argument is X-ray. And then like you have to make sure like you are giving it a good output director. So for here, to update it actually. For here, like we say like I want to write all the preprocessing outputs into this folder. And then like you have to give it the alignments from what you pre-generate, the MSA files. And then the following two is basically for the performance consideration. So DeepSeek attention will give you like better memory performance and also faster iteration. And the max recycling iteration will be used for the AlphaFold 2 model to do the iterations like as you instructed. So ideally, like more iterations will give you better models or more confident models. But the trade-off is like you have to pay like a little bit more time and computing resources for that. So once you have run that, it will generate these output as you directed. If we go into that, you will see like it will generate this rocket input file for you, which is also, as I mentioned here, like, so first it will try to convert the MTD file you provided before with the error estimated parameters involved in the Phoenix one to make it a new like MTD file. And second, it will like, if you don't really have the pre-doc PDB file you provided, it will try to do the MR solution for you, or it will just rename what you got here. And then it will try to convert or like try to align the AlphaFold to initial prediction to the ML solution you give it or it generated before. So it's called predicted aligned PDB. And beside of that, like because sometimes like your notion, like even in a single protein, you have like multiple domains which can have some hinge motions or like decoupled rigid body motions. So this one is a text file which will try to tell you or tell Rocket like maybe there are some like different segments in this PDB, so they'll try to align them separately to make sure the following process is like more smooth. And besides of these input folders, it will also give you two YAML files, which are actually like the configure file required for the following rocket refinement run. And this is what they look like. So it's basically starting from a node. The node is like how you identify this particular run. So we leave a placeholder here for you to write your own node here. For example, I can leave some date here or some like specificity of your targets here. And then like it will show the data mode, what is your R3 flag, what is your test set value for that R3 flag. And you can also set what is the mean resolution, what is the max resolution for cutoff of your MTV file. And more importantly, like it will tell you like what the absolute path you are currently working in. This should be like the pre-processing output files you just generated. And beside of that, it will tell you like what is the initial PDB file Rocket will try to treat it as a reference. So for this one, it will be the predict aligned PDB we just generated using preprocess. And another part is the UUID hex, which is another identifier which appears before the nodes you predefined. This will be used to generate the unique output folders for this ROM. Okay. And then like basically what you can do if you don't need this RK cluster and RK score run, as Alisia mentioned before, you can just directly run rk.refine using these config files. It's pretty simple. But here, for this like SERPing case, as Alisia mentioned before, we actually need a like large fold switch to really move the loop back to the system, as I mentioned here. So this is the AlphaFold prediction, and this is what we really need. So you basically need some like large fold switch to really fold this loop back in. So additionally, we have to run some like MSA subsampling and MSA choice based on the experimental score to really choose a better initial point for Rocket to run. And here is like the example of the commands we use to run this MSA cluster, MSA subsampling, and the following RK score. It's basically trying to give it a name and guide it to the folder with the pre-generated MSA, like the full MSA generated before, which is the alignments folder, as I mentioned above. And this is the name of the output folders for the following subsampling MSAs. And the number control is like how many of the uniform subsampling we want to generate. Like if you set this number to be larger, it means like you'll generate more subsampling, random uniformly subsampling MSAs, but also like in following score, you might have to pay like more computing resources to go through each of them. And this is what it looks like here. So it will try to generate all the MSA files from clustering, also from the random uniformly sampling, subsampling as you guided here. And in the following step, the RK score will try to, using each MSA files coming from the subsampling process we did before, to run the experimental score to say like which one of them will give you the best initial models given your experimental data. And the results will be like, of course, like for each MSA you have one PDB file, but also we will generate a CSV file with the scoring for each of the subsampling MSA we defined before. And we are we will choose that for you, but like also like this ARG column is basically a metric of how good your initial model will be consistent with your experimental data. Larger means better. So in this one, like we will choose the highest one for you, but also you can try to pick some other candidates as you're interested. So for example, for this running case here, this one actually gives us largest error G, which is V1034, as we mentioned also in the manuscript. So in the following run, instead of like directly running the refinement configuration as we in the default mode, we will try to run another refined config file by pointing to this subsample MSA. And how it looks like, it's basically just, it's basically just the same config file as I instructed before. The only difference is like now you have to specify what is the input MSA, because before we are setting it to be none, so it will use the whole, like the full MSA, but here by setting a specific input MSA, it will just start from this subsample MSA to run the following rocket or refinement. And all other things are the same. And if you just simply call like AlQuraishi refine config YAML file, just run under this config, it will run the refinement for you using like different iterations, different number of runs. And then it will generate results inside of this rocket output file or folder, which is under here. Still in the processed folders we defined before. And it is inside this rocket outputs folder. Yeah, and this folder name is coming from the UUX ID we define in the config file, in the YAML file. And this one is actually the nodes you predefine in the YAML file as well. So, and then inside of this, you will have like, first, you have the refinement trajectory PDB, which includes all the PDB files for each iterations, for each set of runs. Like we call it using the alphabet letters like A, B, C. So those one are the ones actually like the videos Alisia showed before. That is actually the refinement trajectory we got from Rocket in this way. And beside of that, we'll also try to pick the best model in all these runs for you by error G value, as I mentioned, which is like a metric between all your models consistent with your experimental target. And this one will be saved as best model, name of runs, and the number of iterations here, which is the PDB file here. This will contains only one single model, which we believe is the best one to describe your data. And beside of that, we also have some saved some like checkpoints of the bias and the weights, which can be readily used for the next future refinement if you are interested. If you don't like really being satisfied with the current model, you can also put it in the phase 2 modes, which you can say like you can specify the starting bias and the starting weights, which actually comes from the best one you got before. And it will try to continue the refinement using the best checkpoint you got before, which is included in the previous running folders. And beside of that, we also like provide the config YAML file, which is used to run this particular run, and all the diagnostic traces which can be useful for your following plot or just like check. And as a result for this run, we can see how it looks like. The best model is like A98. So I'm trying to show it here, which is the model coming from Rocket. This is ground truth. This one is coming from AlphaFold 2. And this one is what we got from Rocket. It actually recover the loop to flip loop back and insert in the correct places. Okay. that is for the X-ray case. And then for the cryo-EM case, it's actually a little bit different, but like mostly they are the same, and sometimes it can be even simple. So if we go to the cryo-EM case, similarly, we have to run the pre-processing first, right? So this is like the data you needed for the pre-processing of Rocket to run in cryo-EM case. So first, you need the map, right? So here, by default, like Rocket will try to ask you for both half maps because like the disagreement of the half maps actually provide some information about like which part of your experiment is more reliable or not. So by default, we'll ask you for the half maps. But if you don't really have that, you have only one single map, you can also like only put one map there. And we also like, I mean, in this settings, as before, we just basically assume like you have some pre-docked PDB files, which will be used as reference in following. But ideally, like if you don't really have that, you can also just don't put that anything. And Rocket preprocessing will try to call Phoenix doc to try to dock initial model for you. But like what is special here is like we actually have multiple chains in this system, and we're only trying to refine one of them as our target protein chain. And the issue here is like we have to, I mean, currently Rocket can only work with one protein chain at the moment. So we have to make sure like your product PDB or like your FASTA file only contains the sequence for your target protein. And at the same time, you try to make a pre-docked protein complex with all the other chains in the systems into this PDB. They don't have to be perfect. They just need to be there, like approximately right. You can use some like other docking tools from Chimera to do that. So this one will actually include all the other chains of this complex, and then they will serve as the fixed models in the whole process. And similarly, we also need the pre-computed MSA files to generate the following predictions. And for the cryo-EM cases, you can also run Rocket preprocess. Similarly, you need the file ID, which is the name here. And one thing special is like you have to provide some resolution of your data, which can actually come in from your like how you generate these maps from like the like the Fourier shell correlation in your pipeline to generate these maps, and that will be put here. And of course, method will be cryo-EM now, and the same output director argument which will use to be created the folders for the following row, and also like you can pre-assume like give it pre-computed MSA file folders. And if you have, you can provide the pre-docked model, which is a good reference for Rocket to know. So you can treat it as a reference to align its prediction to it as a first iteration. And also like if you have multi-chains in this system, as I said before, you can provide this fixed model with the docked chains from with the docked models from all the other chains. And here, if you have half maps, as I said, you can just call it map 1, map 2 by giving you your 2 half maps. But if you only have one single map, you can just call it -- map and give it the only map you have. It will also work. And those two are the same arguments as I mentioned before, which is required for the underlying OpenFold AlphaFold to run the prediction and make it efficient. And similarly, it will make these folders for you after it finished run, and there will be a Rocket input which it prepared for you. And which include the processed MTZ files from the map you gave. So like we are working with a target in the reciprocal space, the doc solution, and the predicted aligned PDB solution. We also like try to convert the PLDDT to B factor, as Alisia said before. And SegGuide is a similar thing. If you believe if the model believe there are multiple domains in your protein. And similarly, it will generate the 2 YAML config files for you as a starting point. So as you can see here, it will try to similarly cryo-EM free flag. It's not used here actually, I think. Include PDB will be the predicted aligned one as we mentioned before. And all the things are similar. And as I mentioned before, like similarly, you can also like do the continuous run after phase 1 run is finished. So we also try to generate this phase 2 run for you, or phase 2 run config file for you, which actually changed input PDB from the predict align to the best models in your phase 1 run. And also like it will like predefine or like make some placeholder for you for the best checkpoints of the models you got from the phase 1 run. And then you can basically just very easily call arc.refine phase 1.yaml, arc.refine phase 2.yaml. And after the running is finished, it will generate, as similar before, it will generate the results in this rocket.output file, which is inside here. This is the UUID you predefined, and this is the nodes you defined. And similarly, it will generate all the refinement trajectory for you, and also try to pick out the best model for you based on the ARG, and other some like diagnosis plots, which can be useful for your following presentation. Yep, that's basically it. And I want to mention like all the examples I showed today are basically more detailed description of the 2 examples we mentioned in the rocket documentations. And also we borrow some of the commands from the launch your own X-ray data and launch your own cryo-EM data, because that is more detailed about how you can pre-process your data. And also like we have made our codes open source in this GitHub repo. And if you got any questions besides of like directly emailing to us, you can also try to raise an issue here. You can say like we have been working very actively on trying to closing other people's issue in raising this GitHub repo. So feel free to leave your issue if you like really try to run Rocket and got into some issues. Yeah, that is basically it.
Yeah. And I guess like one thing I'll add is these config files that Minhwan just showed. Actually, everything. So you can play around with the inputs, but we've made it so that the like defaults that are just generated by Rocket should be good to go. So unless you're specifically looking for something or something is not working right, what you essentially want to change is just your note, right? So like the note that you want to give for the specific run, like maybe your sample file or the specific identifier that you want to call to re-identify the run later. Everything else you should just like kind of automatically work. But we do provide the config files open because like some people might want to be more adventurous and actually try out with different parameter settings. But the defaults should be good to just like naively run it and give it a try.
Yep.
Yeah, so I guess we'll open the floor for questions if there are any.
Yep. Yeah. Thank you both. That was a very good talk. I have a whole stack of questions, and as I was going, some of them you were answering them, but my questions are probably the least interesting. So folks in the audience, if you've got questions, feel free to send them in chat, use the Q&A, raise your hand, and yes, folks are going. So One question from Zong Li. Do you have to type in each command for each step? I would guess yes, but I will defer to you folks.
Yes, so there is you'll have to type in essentially the rk.preprocess command and then rk refine phase 1, and then if you want rk refine phase 2. The difference between phase 1 and phase 2, we kind of explained this in the methods of our preprint, is phase 1 is a bit more like aggressive and adventurous. So it tries to first kind of screen if there are big changes that are needed. Phase 2 tries to then kind of polish into like a smaller well. So usually we would recommend doing both, but it also can depend on how close your initial prediction is to the actual experimentally determined structure. So usually you can actually tell by looking at these like loss LLG runs. You will see if it has nicely converged or if it hasn't.
And so I guess to follow up on that, sometimes people will run through multiple steps in a single script. Is this one where you'd recommend people like pause after each step and look at the results before they move on? Or would it make sense to just like run them all and look at them whenever they're ready?
So maybe I can answer this, Minwon. Like we've baked in a system that essentially, if we have a set like maximum number of iterations for both phase 1, but especially phase 2. But what we have kind of baked in is that if the model is not improving, it won't just keep running. So it won't just keep wasting your GPU time. It will stop. So if it has kind of a self check that if it hasn't improved in 20, I think, iterations, I forget what we converged on, it will just stop. So there isn't really like such a bad downside to kind of automatically running both. Like this is what we've done internally.
Cool.
Yes.
Thank you. All right. I'm going to see if I can unmute Mushan and he can ask his question. I'm trying to unmute you, Mushin. I don't know if it's working or not. Hang on just a moment.
So I was trying to write send my question in the Q&A. Thank you for unmuting me.
Yeah.
Can you hear me right now?
Yep, I can hear you. Yep.
Okay. So my question is because I'm slowly pitching the structural biology thing. I was mostly protein biochemistry guy for my PhD, and so I'm trying to do cryo-EM or crystallization, whatever, and I do a lot of modeling, AlphaFold. But what I summarize from your talk is like, you have an initial sequence, you model with AlphaFold, you get a lot of regions with confidence and less confidence. And with this program, you can actually, refine that kind of molecular similar to maybe molecular replacement. Maybe I'm wrong using that term. And you get less confident region. You actually reduce the less confident region. Is that what you say, like the program is for?
So do you want to go, Minhwan? Sure.
Yeah, I would try to say like when you say confidence, there are actually two terms which you can say as confidence. First one is like the PRDT value predicted by AlphaFold 2 models, right? It's the model self-identified confidence, and the other confidence which is the consistency with the experimental data. So if you're meaning like the confidence, meaning like the consistency with experimental data, I would say like our program is trying to improve that consistency with your experiment data by trying to change the atomic models underlying it.
And I guess I will add, so there is actually a big difference with molecular replacement or docking, which is that in the case of molecular replacement or docking, what you're doing, it's kind of what I showed in my first slide. You take the initial prediction and you try to like place it in your map, or in your density, depending on what data type you're using. But what Rocket is trying to do is actually the next step. So once you've actually like docked that into the map, the AlphaFold prediction itself probably will not actually be a complete reflection or a perfect, well, no model is perfect, but it won't be maybe the best model that actually explains the data, because often the kinase example I showed is a good example where maybe actually AlphaFold's prediction is a really good one, but it's of another state of the protein, and you actually have collected data where the protein is trapped by a drug or by some other kind of environmental condition in another state. And so what Rocket will try to do is like correct that for you, so that you either spend less time doing it manually, or if you have maybe low resolution data and so it's harder to do it manually or with the help of traditional refinement tools, this might help, because it doesn't rely on the same type of geometric restraints that you would normally.
So this can be used instead of CryoSpark. So you have the raw data from the cryo-EM, then you use that here in the Rocket, and you kind of refine that, your initial data. Is that it?
Yeah. So the idea is like you can generate the map, which is the 3D volume voxel intensities from the CryoSpark, and you can use that as an input for Rocket, and Rocket will try to refine the underlying atomic models for you, which is consistent with your map from the CryoSpark.
Okay.
Yeah, if I could jump in there, my understanding is that Rocket is not going to help you go from images to a 3D reconstruction, but then once you've got a 3D reconstruction
Right.
and you'd like to go on
Yeah.
to or make your model better, that's where it'll help.
Exactly.
Right. Exactly. Yeah.
Alice?
Hello, can you hear me?
Yep.
Nice. Great talk. I thought it was really interesting. I also thought your slides were very visually beautiful as well. My question is about oligomers, like heterooligomers, because sometimes you have a structure where the map that you've obtained is multiple proteins in one map. And I'm wondering, how does Rocket handle when you provide it with a docked model and a map that has density for multiple proteins in it? Does Rocket perform well in just restraining that protein just to the density for that one actual protein that you have, or do you need to segment the map and provide Rocket with a segmented map? Because in some cases it's rather fiddly to produce a segmented
Right.
map if you don't already have
Right.
a model refined like into the density,
Yeah. So usually this is maybe even most common, I think, in cryo-EM. So I don't know if you're thinking of cryo-EM examples right now. So this Minhwan touched upon this when he explained the fixed model. So we've accounted for this. So normally the kind of one thing we are relying on is that you've at least like docked some maybe rough version of your proteins in the map. If you've done that, then you're sorted. So say you have a map that has like protein A, B, and C, and you've like you've docked those in Chimera, and then you only want to refine protein A, or maybe you want to refine like first protein A, then protein B, then protein C. At the moment, you need to do it one by one in Rocket. And so if you want to refine only protein A, when you go into this, when you give it this kind of Rocket preprocess command, you can provide this fixed model. And the fixed model is just the PDB of docked proteins B and C. And if you provide us with that model, we internally account for telling Rocket this part of the map is accounted for. You don't need to build in there. And so we've seen that it works quite well. We essentially cancel out the Fourier contribution from that part of the map. So you don't need to segment the map, but you need to give us the kind of rough contributions of what models are in there. And like Minhwan says, they don't need to be perfect. But maybe if you've like roughly docked AlphaFold predictions there, that's already a big help.
What if you don't? So what if I provided just the one protein I'm sure goes kind of here, and I don't provide the others? Because in some cases you can't provide them until, because you're just subtracting like, you know, volumes and stuff. So what would happen if I only gave one protein, but the map for a complex?
So Rocket will still run, and chances are like it will be okay. The only case in which there might be a downside is imagine that you maybe have a situation where you have protein A and B that are very close to each other. And it's maybe it also will depend like on the quality of your data, right? But maybe you have Rocket could maybe get confused because it sees map volume that could perhaps like fit part of the protein, and like move part of like your protein in there. We actually like we're a bit worried about this. We actually haven't seen it happen yet in our tests. So to some extent, if the depending on the radius of convergence of the refinement, you might be okay, but you may not. So maybe that's a situation where if you're particularly worried about it, like run Rocket and just take a look at what happened afterwards, and maybe check that it hasn't gone somewhere where you like wouldn't expect it. Essentially, in that case, the model no longer knows that it should not take that into account. So it might try to take it into account and like think it should also try to explain that volume.
Yeah, okay. That makes sense. I mean, that's what the investigator would be doing manually anyway, right? Like you have a density and you don't really know where your model's supposed to go, so you're just pulling and checking and seeing if is the density continuous, and heaven forbid you have discontinuous density at a couple loops, and then it becomes like a bit of a problem. Actually, that's a good follow-up question. How does Rocket handle if you have discontinuous density, like it turns structures and loops where the density breaks down?
So we've seen it handle it quite well. Actually, this is a part where, the fact that you have this strong prior that comes from AlphaFold helps because, AlphaFold, like compared to maybe when you're in Qt and you try to like drag the loop here and then it falls back, like AlphaFold can say, okay, like I think this could be a good loop. And then it sees how much of the density in that region it fits. And so we've seen that it can help in these cases. Of course, it's not miraculous, right? So it's data driven, and so in a case where there is no data for a loop because the loop is so flexible, the best that Rocket can do is tell you, well, this is a loop that could fit here and that kind of matches the AlphaFold prior. But what we've actually seen is that it kind of will self-report some low confidence for this loop, so it will tell you. I'm not as confident here. And it probably is related to also the fact that it didn't find good feedback from the map in this region. So it can help when there is like poor data, but it's still informative. So it gets rewarded by the loss function for placing atoms in a specific way. If there's kind of no gradients coming from the data, then of course it won't be able to. Thankfully, it doesn't hallucinate as much.
That's good. Thank you so much.
No worries. I should also say, like, so this is kind of we're just making Rocket available. We're just making it public, and so there might be behaviors that we run into as like more people try it that like we may not have accounted for or. That we personally haven't foreseen. So if you do run into something funky or if you run into something strange, like please just like Minhwan said, like write on the GitHub. We keep a close eye on it so that we can try to, as fast as possible, kind of iterate and improve any glitches that become apparent.
We still have a couple questions coming in. Are the two of you okay to stick around for a couple more minutes to answer questions, or do you have to run?
No, we can stay.
Yeah, we can stay.
Awesome. Thank you. So I guess one of the follow-ups is kind of a transition on the what we were just asking about and the partial models. How do does the VRAM requirements scale according to the non-fixed portion of the model or according to the total model? And I guess partially where I'm going here is I'm wondering if it would be possible to cut down the VRAM requirement by faking out a smaller region of your complex and say, just refine chain E, and maybe that will fit in the available VRAM, whereas the entire complex might not.
I guess like by VRAM, you mean like the GPU memory
Correct. Yeah.
requirement, right? Yeah. So like the basic idea is like most of the time you need the full sequence to predict the correct fold, right? So that is a really time-consuming part because you have to. So there's like attention kernel in this AlphaFold 2 model, which is the most memory intensive. So if you already put the full sequence into the model, it will just at the end it will consume that amount of memory. So like even you cut off the model at the end or cut off your map at the end, it will not really solve the attention kernel memory requirement part. That is my quick answer. But of course, like because of you cut off the map, because of the cut the model, the computation from the likelihood part will save some memory, but we don't believe that is a major bottleneck of the memory part.
And I guess the kind of follow-up there is we have not tried to optimize the memory at all up to now. We've like focused on the density and kind of speaking with the OpenFold team here, we actually do believe that there are some gains that we can implement in the next couple months. Just because there are some low-hanging fruit that there was never any reason to optimize that we now have a reason to optimize. So we do hope that there will be an improvement there relatively soon in the relatively near future.
Cool. Yeah, I mean, you always want to make it, do what it wants to do or do what it should do before you try to optimize it. But yeah, 80 gigs of VRAM jumped out as there are a lot of people that don't have that much VRAM hanging around on their systems.
Right, and again, hopefully we will have a server available as well that's in preparation in collaboration with the Phoenix team that will kind of enable people and users to try it even if they don't have locally available GPUs.
Cool. I guess so. Related to that, there's some of the machine learning toolkits have tried to be cross-platform to support Apple with the unified memory versus just strictly CUDA. Is that something that is potentially an option for Rocket, or is that just still everything is CUDA and worry about the rest later?
So I think now everything's CUDA because like we depends on OpenFold. But I think like in the next version or like the possible involvement of the OpenFlow part, like they might to involve AMD or like even Apple Silicon support in some sense. But like again, like because Rocket requires to take gradients, so maybe they can be faster in the inference part, but like they might not be working that well in really the Rocket pipeline. But I think there is some room for improvement there. That's my bottom line.
Cool. I guess so. Jumping two related questions. Do users have to input the sigma level of the maps? And when you're inputting the cryo-EM maps, are these the filtered or sharpened maps, or are these the raw ones?
So sigma level, is that what you said?
Correct.
No, so all we need actually for the crystallography case, we don't even need the resolution because we do an internal resolution test in the pre-processing stage. So that part, no. The answer is simple, no, and we don't need the sigma. We don't need the sigma levels. So we take care of this on our end, like thanks to also the help that we've gotten from like Randy Reed and Early McCoy from Phaser and Phoenix. The unsharpened sharpened map. So we've worked with a few users on this. Ideally, you would provide the two half maps. And this is mostly because we do like an error analysis, looking, like Minhwan said, at the kind of correlation between the two half maps. And so we would prefer, like, we prefer that in terms of how we can account for like the error. And so maybe like not focus on specific regions where we know that the signal to noise is slower. That said, if you have a post-process map or a filter sharpen map that you like really believe could be just like a better map to build to, then what you can do is provide it as Minhwan said, rather than do like map 1 and map 2, you can just do dash map and provide us with the map, with whatever map you prefer. If it's in MRC or map format, it will work. And so it will be fine. I guess we just tend to encourage giving us the half maps because of the internal error analysis. But we've recently worked with a group that gave us a post-process AI post-processed map, and it worked really well. So it's definitely an option.
Does it matter if if a user specifies the half maps and that those half maps have a certain FSC resolution, but they give you a different resolution on the argument? Does that cause confusion or chaos, or is this something I should get up a shoe about?
Yeah, it will be a marginal, like unless there's a huge gap, it will be probably actually a marginal effect. What it might lead to is that we include data, say we include data kind of like maybe past the true resolution limit of your data, and so like maybe you have some extra kind of noise coming in, or vice versa. I don't think it would make a huge difference if you have like a slight mismatch between these two numbers.
Thank you. I think one more, and this may be the last one, is if the AlphaFold model is not necessarily folded well, like for example, if the true state has multiple confirmations, like how well does Rocket handle that? Or I guess how does it handle that?
So is it if it's not folded well, or if there's multiple confirmations in the
Yes,
data?
that is actually two separate questions.
Yes. We can answer both. Perhaps I can answer one, and Minhwan can answer the other one. So the first one, like what if the AlphaFold model is not well folded at the beginning? So there are a couple kind of like reasons why that might be, and that will maybe impact how Rocket can help you recover. So the first is if the prediction is bad because there is no coevolutionary information. So say you don't have an MSA, or you have a very shallow MSA. Then here, we haven't tested this extensively, but we don't expect that Rocket will be able to have huge improvements because, as I kind of outlined, like the key concept behind Rocket is that we optimize and we kind of bias coevolutionary information. If that initial matrix that AlphaFold has is full of zeros and it has no information content, it will be hard for Rocket to probably really recover a confirmation from this. Like if AlphaFold is really struggling, then that kind of structural prior that we rely on is not available. If the initial prediction is so bad that a molecular replacement solution is not possible, and so we can get no gradients from the map, that of course also puts us in a difficult, like Rocket is unlikely to help because it needs kind of a true signal coming from the data, and if the model is not docked or molecularly replaced, it might just not give you meaningful results. If it's an in-between, so if you do have like a reasonable MSA and the initial structure is, AlphaFold is unsure of like a specific loop position because it has seen multiple loop conformations in training, but the data that you have like is strongly preferring one of these, then you like Rocket might be able to help. So there's kind of different reasons, different scenarios for that question. I'll let Minhwan answer the what happens if there's multiple conformations. It's just at the
Yeah, so like
moment.
yeah, at the moment it's currently like because now you can imagine like Rocket as a point-wise estimation or point-wise optimization approach. So to only give you like one single models for each run. But the thing you can do is like you can start from random like different random seeds or different initial points and hope it can give you different results, both consistent with with the data at the end. Or you can try to like maybe do something like, for example, in X-ray, you can try to do some extrapolated approaches, try to maybe like bias your map to one of the two states you believe it exists from the beginning, right? So that is one approach currently maybe possible in Rocket. And the other approach which we're currently working on is basically instead of like trying to think about the approach as like point-wise estimation, we're trying to really sample from the posterior given your data, which is then becomes like ensemble sampling problems. And that is becomes like even possible with that like more the next version of the AlphaFold. And that is something we're trying to working on now.
Yeah, so that's not available at the moment, but it's something we're working on. So the automatic fitting of multiple structures at once. And of course, there are considerations like in doing this properly. And so we want to make sure that we don't overfit and that we're still like accounting for all our experimental errors. So that will probably become available a little bit later.
Cool. Thank you. I think
Great.
we may have run through all of our questions other than me asking like, hey, this is similar to this other cool technique, and this is similar to this other thing, and mathematically this is interesting, and we're already over time, so we shouldn't talk about this. I guess one last call for like anybody has any questions. All right. Thank you very much for joining us and presenting and for sticking around and answering all the questions. And look forward to seeing the future developments.
Thanks so much. Yeah.
Thank you. Thank you so much.
And just get in touch on the GitHub if like any questions come up post. Bye.